Good morning, I'm Elise Modica, and here's the latest update from Spectrum Autism Research. Autism Gene Interference Silences Song Memory in Birds by Sarah DeWeert. Muffling expression of an autism-linked gene in young zebra finches' brains renders them unable to learn songs from older birds, according to a new study. But it does not alter their ability to practice and perfect previously memorized melodies. The gene, FOXP1, belongs to a family of genes that regulate the expression of other genes and shape nervous system and language development. People missing one copy of FOXP1 have language impairments and intellectual disability. Those with mutations in the gene often have autism traits. Our overall goal was to try to understand if we could use songbirds as a model system for understanding how genes associated with autism can influence the transmission of behaviors from one generation to the next, says study leader Todd Roberts, associate professor of neuroscience at the University of Texas Southwestern Medical Center in Dallas. Songbirds such as zebra finches learn to sing by imitating adult birds, much as young children learn language and social skills by mimicking their caregivers, a process disrupted in autism. This learning involves forming a memory of a song motif, a word, or a social behavior, such as waving, and then practicing that memorized riff or action to perfect it. The new study suggests that FOXP1 plays a role in the first step of that process. It shows that we can start to disentangle these different aspects of learning, Roberts says. Other research has found that turning down the activity of another autism-related gene, CNTNAP2, in song-related brain regions also impairs zebra finches' ability to learn songs. FOXP2, a gene closely related to FOXP1, is also involved in song learning in the birds. But relatively little has been known about the role of FOXP1 in vocal learning, says John Cicada, associate professor of biology at McGill University in Montreal, Canada, who was not involved in the work. I think the experimental design and results are pretty compelling with regard to learning. Timing tips. Roberts and his team designed a molecule that partially blocks expression of FOXP1 and injected it into the brains of male zebra finches at about 35 days old, an age at which the birds are typically engaged in memorizing songs. The researchers aimed the injections at an area called the HVC, which roughly corresponds to certain speech-related regions of the human cerebral cortex and plays a key role in song learning and production in songbirds. Cells throughout the HVC express FOXP1, especially neurons that connect with another song-related brain region called Area X. Birds treated with the FOXP1 blocking molecule had about half the normal amount of FOXP1 protein in these neurons. Roberts and his colleagues expected that this dearth of FOXP1 protein would hinder the practice and perfect phase of song learning based on past research. But birds that had heard adults singing before FOXP1 was disrupted grew up to sing typical zebra finch songs, the team found. That was really surprising to us, Roberts says. So the researchers turned to zebra finches raised in isolation without the opportunity to memorize adult birdsong. They injected these birds with the FOXP1 blocker and then put them in a cage with an adult tutor. It was kind of like a last-ditch effort on the project, Roberts says. And then that's when we saw this huge effect. The birds that had FOXP1 expression dampened before hearing a tutor's song grew up to sing variable and disorganized songs, much like those of adult birds that never heard adult birdsong during their youth. The results suggest that FOXP1 has a very specific effect just on the ability to encode memories of the social model that you want to imitate, Roberts says. The study appeared February 3rd in Science Advances. The new study does not address the role of FOXP2 in the song memorization process, says Gudrun Rappold, a geneticist at the University of Heidelberg in Germany, who was not involved in the work. The proteins encoded by FOXP1 and FOXP2 form complexes with each other, and both interact with CNTNAP2. Future research should aim to tease out how FOXP2 interacts with FOXP1 in the HVC when birds are forming memories of a tutor's song, Rappold and Cicada both say. Bursting block. Roberts and his team also investigated how dampened FOXP1 expression affects brain activity when birds hear a tutor's song. The shortage of FOXP1 protein made neurons linking the HVC and Area X less excitable than normal, they found, whereas these neurons typically become more excitable when a bird is first exposed to a tutor's song. It also disrupted the organization of neural activity into bursts, something that typically occurs when the birds first hear a tutor's song. Exposure to adult birdsong also normally increases the abundance of AMPA receptors relative to NMDA receptors on HVC neurons. But this shift did not occur for birds exposed to the FOXP1 blocker before hearing a tutor's song. Finally, the scarcity of FOXP1 slowed the turnover of dendritic spines, protrusions involved in receiving messages sent between neurons, a process that is known to help birds learn tutor songs. These are just all very classic, strong correlates of learning and memory formation, and are all disrupted by the loss of FOXP1, Roberts says. Dampening FOXP1 in the HVC doesn't affect only neurons that connect to area X, Sakata says. So other HVC neurons could also be important for encoding song memories, as could FOXP1 in other brain circuits. For example, interfering with FOXP1 in area X also impairs song learning in zebra finches, according to their research. Roberts plans to conduct similar experiments in other brain regions involved in song learning and song production. To listen to the bird calls, go to spectrumnews.org or click the link in the show notes. That's all for today. Check back on Friday for more content from Spectrum Autism Research or go to spectrumnews.org.